And welcome today for the latest session of this webinar entitled Spheroid Encapsulation in Alginate Microbeads using Microfluidics. I'm Joseph Farah, Microfluidic Application Engineer at Fluigent, and today I'm pleased to welcome Dr. Adrien Dewandre, co-founder and technology lead of Sequoia Technologies, and Dr. Léon Rambot, who did his PhD at the Bioimaging and Optofluidics team of the Photonics Lab affiliated to the University of Bordeaux. So to start, we'll start with Dr. Adrien, who will present us first the technology, and afterwards with Léon, the part of the how to encapsulate and generate. First, how to generate the alginate beads, and afterwards encapsulate the spheroids using the microfluidic techniques that we would like to present. Adrien, the floor is yours.
Thank you, Joseph. Okay, hello everyone. I'm Adrien Dewandre, co-founder of Sequoia Technologies. I'm very pleased to participate to this webinar organized with Fluigent and Léon Rambot from the Université de Bordeaux. I will first give you a short introduction about our emulsification technology, and then I will let Léon talk to you about the main topic of this webinar, the encapsulation of cells in alginate microgels. So to encapsulate a molecule or a cell in a particle made of alginate or another polymer, you first need to produce an emulsion of one liquid in a non-miscible other liquid. In the standard batch process, fast stirring is commonly used. And then after the cross-linking process, depending on the formulation, a solid or gel particle is obtained. The particle are typically present a large size distribution and a poor sphericity. And what is not ideal, of course, for a precise loading, and of course, this technique of fast stirring is not compatible with cell encapsulation. On the contrary, using microfluidics tool, you can directly produce droplets with a very narrow size distribution, a tunable size by changing the flow rate in the system, and a high sphericity. And this process is a milled process, so it is compatible with cell encapsulation. Okay, now let's have a look on what tools are available to produce droplets in microfluidics. A planar chip with channel engraved in glass, PDMS, or plastic are commercially available. I used them intensively when I was in an academic lab, and I was at the time confronted with their limitation. Because of this planar geometry, the droplet phase is always in contact with the top and the bottom of the chip, glass or PDMS, leading to wetting issues. And to prevent these wetting issues, you need to apply coatings on the channels, hydrophilic or hydrophobic, depending on water in oil or in water emulsion. And the drawback of these surface treatments is that they are not robust. With the formulation I tested at the time, the hydrophobic coating resisted approximately 20 minutes, and then I could throw the chip away. Another option developed in academic lab rely on the alignment of two glass capillaries in a third capillary, square rounded capillary. This device don't need coatings because of the cylindrical geometry of the system, so the droplet phase is never in contact with the extraction capillary because it's surrounded by the continuous phase all around the droplet generated. They also permitted to produce double emulsion, a drop in a drop, but they never reach the market because the fabrication process is difficult to be reproducible. So what was missing was a device able to produce double emulsion with a reproducible fabrication process, standard connection using HPLC connection type that you can easily fit to the system to connect the pumps and to collect the droplets, and a system being robust and versatile. So not with no coating for both oil in water emulsion or water in oil emulsion. That's why we developed a glass capillary device, a robust glass capillary device called the Raydrop. And I will show you now a small, short video to explain you how this device works. So this device is for the production of single and double emulsion. It is made of 3 parts, a main chamber in metal and 2 inserts in metal inserted on the lateral sides of the main chamber. On one side, you have a glass capillary with a 3D printed nozzle at the tip of this glass capillary for the injection of the droplet phase. And in front of this 3D printed glass nozzle, we have an extraction glass capillary. We first connect the continuous phase to fill the main chamber under pressure. Then we connect the continuous phase, the droplet phase to the nozzle tip, and we connect an extraction tubing to extract the droplets transported by the continuous phase. At the junction of the nozzle and the extraction glass capillary, the jet flowing out the nozzle is stretched by the continuous phase, leading to the formation of very reproducible droplets. Now, if you want to produce a double emulsion, you just need to replace one part of the system, the injection insert, by an insert able to carry 2 phases, the core phase and the shell phase. And in the nozzle, at the tip of the nozzle, the core phase is surrounded by the shell phase, leading to the formation of a very monodisperse double emulsion. So using this system, you can produce single emulsion and double emulsion in one step without needing to apply coating in the system. And this Raydrop is connected to all of the equipment with standard HPLC type fittings. So easy to be connected. So now we have this robust droplet generator, but to produce and control the droplet formation, the Raydrop, as I said, needs to be connected to a lot of equipment, pumps, valves, flow meters, camera, etc. And if you are not an expert in microfluidics, this setup and the control of the emulsion can be complicated and time-consuming. And if it's not your focus, it's a waste of time, I would say. So to overcome this, we developed with Fluigent this platform that you see on the slide that integrates all the instrument, the equipment needed to operate the Raydrop. On the right side of the instrument, you have the mechanical and optical parts, so camera, lamp, and translation tables to support the Raydrop. And on the left side of the equipment, you have the fluidic parts. And you have 3 pumps. We have three Fluigent pressure controller pumps to control the fluid in the Raydrop. With this pressure controller, we apply a very precise gas pressure on the reservoir to push the fluid in the system. Compared to a syringe pump, this technology of Fluigent offers greater stability in flow control, and most importantly for this application, a very fast response time. So if you adjust the pressure, the flow rate will adjust immediately, which is not the case with syringe pumps. That is very important to obtain a stable double emulsion. To demonstrate the versatility of our system, we developed a series of applications on single and double emulsion using formulations widely used in publication. And now we will focus on the last application we published in Labo de Chip in collaboration with Université Libre de Bruxelles and Université de Bordeaux on the encapsulation of mammalian cells in alginate microbeads. Léon, the floor is yours.
Thank you, Adrien. Okay, so hi everyone. I'm gonna describe the work we did with in collaboration with Sequoia and my previous lab at University of Bordeaux. And I will present to you this paper we published recently in the beginning of the year in Lab on a Chip. And I will show you what we can do with this Raydrop, how we use it to encapsulate cells and allow them to form multicellular spheroids inside alginate microbeads. So first, a very quick introduction to why why all this work and why the motivations for these projects. So a quick introduction to multicellular spheroids and more generally 3D cellular aggregates. So spheroids are, simply put, a ball of cells, which can comprise a few hundred to a few thousand of cells and measure typically a few hundred microns in diameter. This is a model that has been around for decades and which has been widely used to model the evolution of tissues, the self-organization of tissues, and especially the evolution of pseudo-tumors that we form in the lab when we put cancerous cells together to form a tumor spheroid. So there's applications in fundamental research, but also a wide range of applications in pharmaceutical industry, where we can use these spheroid models as alternatives to 2D cell cultures and animal testing for drug discovery. Okay, so the way you can form these spheroids is by preventing cells to adhere from the substrates of the culture dish you put the cells in. So by preventing them to spread, you force them to come together to self-assemble into these spheroids. So the historical technique that was developed a century ago was to simply pipette cells into drops of culture medium on a cover slip, then just flip the cover slip upside down, let the cells fall at the bottom of the drop, and this would allow them to form these spheroids. So now there's been evolutions to this technique that allow to form cell spheroids inside microwells with low adherence surface. But more importantly, in recent years, there's been many developments in encapsulating cells inside hydrogels, whose purpose is to recapitulate the properties of the extracellular matrix in the body and to allow for the production of spheroids more closely to physiological conditions. So in particular, one hydrogel that has been widely used is alginate. So again, a very old publication. So as alginate has been around in the biomedical field for decades too. Alginate is a natural bioinert polymer with which the cells cannot interact at all in its native form, which means cells do not degrade it and they are not affected by it and they proliferate freely inside alginate hydrogels. So alginate, we can find it in solution, in a very viscous solution, and we can cross-link it by simply putting it in the presence of divalent cations like calcium or barium chloride, for instance, which will just cross-link the chains of the alginate and form a gel with controllable mechanical properties. One of its advantages, beside being completely biocompatible, is also the fact that it's completely soluble in classic cell culture buffers such as PBS, which means we can form spheroids within alginate microbeads, dissolve the alginate to recover just the cells for subsequent analysis. So the main problems with current methods to form spheroids, the most widespread ones, is that they rely on the fact that the operator would pipette cells and alginates inside a calcium bath to form droplets one by one and allow them to reticulate. And this is the very straightforward approach, but it's very time consuming. And this limits a lot the applications we can have because we cannot reach the high throughputs of production that are required to in applications such as drug testing. So this is precisely why microfluidics come into play. So instead of taking a pipette to form these microbeads, we can simply use it's a big one, but we can use microfluidic channels to form water in oil emulsions or alginate in oil emulsions that contain cells. And then as soon as we form these emulsions, then we need to bring the divalent cations in contact with alginate to cross-link it to allow subsequent recovery of the microbeads. So there's two approaches to do this. Either we first form a single emulsion with alginates and cells inside oil, and we fuse these emulsions with other emulsions of containing calcium or divalent cations, or we can also collect these simple emulsions into a bath of calcium. So this is the simplest approach. the problem with this is that you may be able to run it for a few minutes in your lab, but usually because cross-linking occurs on chip, any disturbance in the flow would allow alginate and calcium to react in the chip and to clog the chip. So these cannot be used for long-term production and cannot be run for hours. Also, because the cross-linking happens instantaneously when we put the calcium ions in contact with alginate, reticulation is not homogeneous, and cells do not live in a homogeneous 3D environment, which can influence their behavior. So the other way we could form alginate microbeads is to directly put the calcium inside the alginate solution, and to use a calcium which is chelated by chelating agents such as EDTA or CO3 nanoparticles. And once the calcium is loaded inside the microbeads, we can release this calcium by changing the conditions from the outer solution. So for instance, if we drop the pH, we can release calcium from EDTA and cross-link the microbeads by the inside, which leads to homogeneous reticulation, but is usually associated with quite harsh conditions on the cells, and it does not allow long-term cell culture inside the microbeads, and it's associated with very low viability. Both these techniques, in addition, have the problem that once we form the microbead in oil, it can be tricky to recover the microbeads from oil, and you need to do several steps of medium exchange to recover the microbeads and go on with downstream processing. So this is mainly the reasons why 3D cell culture in alginate microbeads is still limited to purely microfluidics oriented lab, and it's hard to transfer this to more biology oriented labs due to complex experimental setups and procedures that currently slow down this field. So this is precisely where the Raydrop chips come into play, and I will leave the floor to Adrien to explain the technology we used in this project.
Yeah, thank you, Leon. Now, as Leon just said, there are single emulsion processes for alginate beads, but they rely on a water-inert emulsion, and at some point, the oil must be removed to resuspend the alginate beads in an aqueous solution if they are to be used for cell encapsulation. So this step is often dirty and quite complex, and plus you need high volume of oil with expensive surfactants to guarantee the stability of this emulsion. So that's why at Sequoia we developed an alternative process to produce this Alginate microgels based on a double emulsion. I know that it seems counterintuitive to work with a double emulsion to at the end obtain single particles, but in this process there is a trick, and the trick is a sacrificial or layer that will protect the alginate from the water of the continuous phase during the gelation process. As you see on this scheme, in this process, we use alginates with cells in the core phase. Oleic acid, so oleic acid is the main compound of olive oil, so it is biocompatible. We use this oil as the shell, so the sacrificial layer, and water with PVA, polyvinyl alcohol, as the continuous phase. PVA is a surfactant to stabilize the emulsion, to avoid to have coalescence when we will collect the double emulsion. So the double emulsion produced in the Raydrop with alginate core, oleic acid shell, and water continuous phase will be collected in a bath with calcium chloride, and this calcium chloride which will diffuse through the oil shell, causing the alginate to shrink and gel. And this process is quite slow. It will take a few minutes. Then when the particles are solids, they can be easily separated from the oil by a gentle centrifugation and collected in suspension in water. And at this step, we can easily pipette the microgel in the bottom of the vial. The oil is on the top, and resuspend them in a cell culture media. So there is no need to perform washing steps or to other procedures with acid or specific oil that could compromise the cell viability. And now I will give back the floor to Leon to show you the results you obtained encapsulating mammalian cells using this process.
Thank you. So the The first argument in favor of our method is that we are able to produce very homogeneous and very monodisperse alginate microbeads. And this you can see here on this graph. If we use FeedC alginate, so fluorescent alginate, this allows us to check whether the reticulation is homogeneous or heterogeneous. And what you see here in the light curves is that we have a parabolic decay of fluorescence intensity as we go from the center to the edge of the alginate, which means that because there is more alginate in the middle and less alginate in the edges, we should have this decay. Whereas if we have, as in the case of the dark curve, if we have heterogeneous reticulation, for instance, because in our case, if we voluntarily break the double emulsions to allow immediate cross-linking of the alginate, then we will fall in the regime where we have the dark curve, where we have a plateau of intensity, meaning that actually there is more reticulated alginate on the edges than on the sides. So in our case, we show that here we have homogeneous alginate concentrations, which is what you want if you want to go further in detail in characterizing the response of cells in a precise and well-calibrated mechanical environment. In addition to being homogeneous in reticulations, the microbeads are also monodisperse. So if we look at the circularity of the microbeads, it's near perfect when we do not have cells. It slightly drops, but almost not without us being able to see it when we add cells inside, because the flow is necessarily a bit disturbed when we add cells, but we still have almost perfectly spherical microbeads, as you will see in a minute. And this is all because we have this slow mechanism of gelation that relies on the diffusion and the transport of calcium through the oil shell. So as you can see here, the microbeads are indeed almost perfectly spherical. And one of the main advantages of these techniques, of our technique regarding cell culture, is that usually when you use, for instance, internal reticulation of alginates with calcium EDTA, for instance, what we get is we have initially a good viability, which decreases over time because cells have been disturbed too much during the encapsulation. But here, what we get is that we have an increase of cell viability over time, simply because, of course, there will be a few dead cells in the process, but they are quickly overtaken by all the live cells that multiply in our system, which means that usually viability is not the only thing you want to check when you do encapsulation, because in many cases, cell will survive the encapsulation, but not long-term culture. Here, we are able to keep the cells in culture for more than a week and to form these multicellular spheroids that you can see here already from day 6 and in day 10. So if we look at many more cells, what you see here is that initially, during encapsulation, since cells do not migrate in the alginate gel, because it's bio-inert, cells are encapsulated and are individually encapsulated in different parts of the microbeads, but each cell that was encapsulated divides and eventually forms a multicellular spheroid, which you can see here at the image on day 7. So if we look closely at what happens inside the microbead, so this is a zoomed version of a time-lapse video of one cell that was encapsulated alone in a microbead. And you can see here that it indeed grows. And we check that the growth rate was exactly the same as in 2D culture here for our cell type, which is, in this case, HEK cells, which are widely used for viral particles production in biology labs. So at first sight, it might seem that it's just one cell that just grows and grows, which is not very possible. And I can reassure you that actually we have several cells in our spheroids, as you can see here in this confocal image of the nuclei and of the actin cytoskeleton of the cells in a spheroid that we produced in our microbeads. And what you can see here is a ring of actin, a shell of actin, which is all around the cells, which indicates that which is a marker of growth under confinement, which is exactly what we have in the alginate microbeads. So one interesting thing we found is that initially the cells are growing perfectly isotropically, as you can see on the first images in the top. We have nice, perfectly spherical spheres, but at some points, the stress generated by the deformation of the alginate due to cell proliferation, this stress forces the cells to grow preferentially in one direction, and we have a loss of circularity over time, which is a marker of this constrained growth. And this would make this system a reliable platform to study, for instance, growth of tumor spheroids under confinement, which is a very active field. So again, as you can see here, this is just an image of one microbead where you can see small aggregates being perfectly spherical and large aggregates being more with more ovoidal shape. And this is just, sorry for the zooming, this is just an image of a cell being dividing in our microbeads. So this was all done with HEK cells, which is a widely used cell type, but also a very robust cell type. So this made things easier, but we then checked with other cell types, such as MDCK cells, which are widely used for epithelial tissue modeling, and also HIPSC cells, so induced pluripotent stem cells. They both passed the test of being completely alive after encapsulation, meaning that our general technique is compatible with encapsulation of much more fragile cell types. We also managed to get spheroids with the MDCK cells, and of course, stem cells are a bit more complex to work with, but what we wanted to show here is that even if you use native alginate, which is not functionalized, which cells do not adhere to, and which is not, in this sense, completely ideal for the growth of complex cell types, we still managed to encapsulate them, and with developments on the matrix, if with a bit of functionalization of the alginate, we could grow virtually any cell type we like. So in summary, in this work, we developed a new method for alginate microbead production, which leads to perfectly homogeneous alginate microbeads and perfectly monodisperse microbeads. This is adapted to the encapsulation of mammalian cells. We also encapsulated yeast cells, which grew completely fine in the microbeads. And more than just being compatible with viable encapsulation, it's compatible with long-term cell culture and formation of multicellular spheroids, which can subsequently be used for drug testing or any application you like. So I will just to finish, recapitulate what happens in this system. So we have these Raydrop chips that allows us to form alginate microbeads with liquid alginates. We collect these beads in a calcium bath. Calcium ions cross a very thin shell of oil, cross this shell of oil, diffuse through the alginate, and reticulate it from the inside slowly, which leads to a homogeneous reticulation. Then the oil de-wets spontaneously from the alginate microbeads. We have a separation of the oil phase and the microbeads, which allows subsequent collection of the microbeads easily with just a pipette, and we can run these experiments quite stably for tens of minutes. We could go for hours, which allows us to form large quantities of cell-laden aggregate alginate microbeads, which will subsequently lead to multicellular spheroids production. So we'll be answering questions in a minute, but if you have any technical detail that you would like to check in the publication, I added the reference there. It's in Lab on a Chip. And with this, I would like to thank you for listening and thank all the collaborators from labs that worked on this project. Thank you, and I give the mic back to Joseph. Thank you.
Thank you, Léon and Adrien, for this great talk and explanations about this method. So now we can switch to the Q&A session. While you are writing your questions, I would like to present quickly Fluigent and the collaboration with Sequoia Technologies. So Fluigent is, we are a French company based in Paris with expertise in controlling the flow at a micro scale. We have a subsidiary in the US and a wide network of distributors throughout Asia. And it's been our 20th anniversary this year since the first flow controller sold and the creation of the company. It's been a few years now that we collaborated and we combined our expertise with Sequoia, our flow control expertise with their emulsion and droplet generation efficiency with the Flow Easy and the Raydrop, either single and double emulsion. And you can check if you are interested further in the applications that we have on our site with the detailed protocol, either in single or double emulsion from the Alginate BLGA, the microcapsules with the chitosan, PMMA, PEGDA, and so on. Or if you are interested in encapsulating biological material or other soluble or insoluble API. So with this, I would like to thank again Adrien and Leon for your time today and the explanations that you brought to this session and the insightful informations. And now we will start with some few questions that we will have from the participants. So the first question for Leon, how many cells you could aggregate at one aggregate? Or like one spheroid, I would say.
Yeah. So for this here, we restricted to beads that were roughly 300 microns in diameter. And in these beads, we had, I would say, a few hundred cells in the aggregates. This depends on what you like to do. Here, we encapsulated approximately 20 to 30 cells per microbead from the start. Each of them starts to form a spheroid, which sometimes can merge, and you have several hundred cells. But we can also go through to single cell encapsulation. They have enough smaller aggregates, so this is quite tunable in a range where you cannot put thousands of cells at a time in the microbeads, because that would just clog the nozzle. But there is a safe range of a few. You can start with a few tens of cells at encapsulation.
The next question, if you have made any experience with CAR T cells, and if you, if yes, like if you could elaborate further on this, on the cell viability.
Yeah, no, this we haven't done so far.
Okay. Another question regarding the smallest or the range, I would say, let's make it more general, the range of the droplet size that the Raydrop can produce.
Yes, with the Raydrop, depending on the nozzle and the extraction capillary you use, you can go from 25 microns to 400 microns in size. And to go from one range of size to the other one, you need to adapt the geometry of the system. And by geometry, I mean the nozzle, the size of the nozzle, and the size of the capillary in front of this nozzle. We offer different sizes of nozzle to be able to reach these different ranges of sizes. And for this experiment, we focus on approximately 300 microns because it is suited for this application. But it should be possible to adapt this protocol to produce smaller microgels.
Thank you, Adrien and Leon. The next question is, in one of the slides that you presented, Leon, there's the bright field picture of one encapsulated cells that grow into a spheroid. It seems like the alginate shell stretch as the cells are proliferating. First, is that correct? And if you can explain further about this point.
Yeah, thanks for the question. Nice observation. Yes, so this is actually correct. So we attribute this to there is actually an exchange in liquid from the alginate and the outside medium due to osmotic small imbalances. So the thing is, we form the alginate microbeads by dissolving alginate in sorbitol at 300 millimolars, which is not exactly the osmolarity of the culture medium. So we used standard culture medium, which is DMM. But any imbalance, small imbalance in osmolarity inside and outside the drops would make the droplets, the microbeads, shrink a bit, which didn't alter cell proliferation at all. But this occurs only in the first, I would say, few days.
Thank you, Leon. A question about the setup, I would say, which method is real-time monitoring performed and which parameters does it provide? I think I'm thinking here more about maybe the microscope or the fast camera that we are using in order to see directly the generation of the droplets. So in the platform that Adrien showed in one of the slides, there is an optical system inside of the Raydrop where, like, you can see the generation of the droplets, the double emulsion directly. And in this case, like, the advantage is that, like, you can adjust, or, like, if you require some changement, like, you can adjust it directly in time, which is like a big advantage in order, like, to see or what to expect as a result directly from this. A follow-up question is if you were able to remove the oil completely once, especially like when you will have the separation between the shell and the alginate beads.
Yeah, actually, we didn't try to remove the oil. Actually, we just pipetted the microgels in the bottom of the vial, and yes, we succeed to remove quite all the oil. There is sometimes some traces on the outside of the alginate bead, but very small traces of oil. Yes, but we didn't try to wash the microgel. So if you want to remove totally all the oil, it could be possible to add some washing steps to remove all the oil.
And it's very minor traces. I mean, it's almost cannot see them.
Thank you. Have you done any experiments where the rupture of the capsules can be seen or triggered?
So this we didn't see with mammalian cells. What we see is that when the spheroid grows and reaches near the border of the alginate, then the alginate would deform a bit, but we didn't keep them in culture long enough for them to break the capsules. But this happened when we encapsulated the yeast. So yeast proliferates so much and generates so much pressure, then yes, they grow and burst out of the capsules, the microbeads.
Another question, what are the wetted material of the Raydrop?
The wetted material of the Raydrop are the nozzle. So the nozzle is in resin, and yes, and the extraction tubing in front of this nozzle is in glass, but the glass is not in contact with the double emulsion. So the cells are only in contact with the cell in alginate, are only in contact with the glass of the tubing to be inserted inside of the nozzle and in the resin of the nozzle. And there is also a possibility to have a glass nozzle.
Okay. The next question is regarding the preparation and the setup. In which solution the calcium, the CaCl2, is introduced, and how sensible the particles were to the selected calcium concentration?
So
yes, the calcium is in the collection bath. So we produce the double emulsion inside of the Raydrop. So inside this setup, there is no possibility of gelation of the alginate because there is no calcium in the system. So this way we avoid to have clogging of the system. Then we collect the double emulsion in a bath with calcium chloride. And for the concentration of this calcium chloride, that is a good question. We showed that in other publication that it was the typical molarity used in previous publication to have the good balance between the low speed of the reticulation of the cross-linking of the alginate, but not too long to not let the cells outside of a culture media too long.
Thank you, Adrien. Maybe another question. How did you address the sedimentation of cells in the reservoirs or on your microfluidic system?
Yeah, that's a good question. Actually, we didn't use a reservoir to store the cells because of this problem, aggregation of the cells in the bottom of the reservoir. So that's why we use in this process we use a sample loop, a sample injector, HPLC type sample injector that's allow us to introduce a very small amount of alginate cells in alginate in a loop of 100 microliter, and then we push this small volume of alginate with cells in the system. And so the volume is very small. This volume is very close to the Raydrop, so it's quite fast to introduce this volume in the system. And so using this we can avoid this sedimentation issue inside of the reservoir and inside of the tubing from this reservoir to the Raydrop. And by adapting the size of the loop, you can work with, let's say, minutes or tens of minutes of production of double emulsions.
A general question that it was asked, I think, several times, the range of the droplet size, or like the droplet size and the alginate beads, and thus the alginate beads that can be produced with this method and then with the Raydrop in general.
In this case, we used a 3D printed nozzle to produce droplets at the end, alginate microgel of 250 microns approximately, because that's why this size is recommended for cell culture, because if it is bigger, nutrients cannot reach all the cells in the center of the alginate beads. So that is, let's say, the maximum size recommended for cell culture. And it's more easy in microfluidics. You have less issue when you work with let's say big droplets, as in this case. But in theory, with our system, it's also possible to make smaller droplets with different size of nozzles and extraction capillary. It's possible to produce double emulsion from, let's say, 25 microns up to 400 microns. But with this process, we didn't try this process, this specific process with alginate and sacrificial oil with the small nozzle. So we don't. Yes, it is possible to produce smaller alginate microgels, but perhaps some parameters need to be adapted to achieve these results, of course.
Mm-hmm. Mm-hmm. The question is regarding the nozzle, if we are using the Raydrop nozzle. Yes, so basically it is the double emulsion Raydrop nozzle, the classical ones that it's being used, and it's not a custom made.
No, it's a classical nozzle. Yeah, the standard nozzle.
The question also a follow up regarding the wetted material inside of the Raydrop.
The wetted material, the nozzle is in plastic, in resin actually. We have also an option in glass if necessary, but it is not necessary in this application because there is no problem of compatibility between the formulation and the resin of the nozzle, and glass for the extraction capillary. Then at the output of the Raydrop, you connect a tubing of PFA, of other polymer. But it's basically this in contact with the cells and alginate, it's resin. And that's it, because there is no contact between the double emulsion and the extraction capillary.
Mm-hmm. And following this application, can we formulate different gels, such as gelatin or collagen?
Yeah, possibly. All the processes that are described in literature can be using microfluidics chips can be done with our system actually.
Okay. Yeah. With this question, I would like to thank again our speakers, Adrien and Leon, for your time today and the great talk that you had. I would like if you require any further information or requests, you can write us directly, or also by responding to the mail when we will send the recording of this session. Also, if you are interested in any proof of concept, we can offer this option based also after some meetings. We have the option of doing some proof of concept if you are interested in this. So again, I would like to thank you all for your participation today for this webinar in partnership with Sequoia. And I wish you a great day. Thank you all.
Thank you. Bye-bye.
Thank you.